Hey, I'm Dr. Peter Jones, Scientific Advisory Board member with FSHD Global Research Foundation, and I'm coming to you from the University of Nevada, Reno School of Medicine in Reno, Nevada, in the western part of the U.S., and that's why I have this wonky accent. And today I'm going to address one of the most common questions we get from FSHD patients, families, researchers, and clinicians, and that is, what is the difference between FSHD1 and FSHD2? And we'll even address what we know about FSHD3. Okay, so put together this little presentation for y'all, and hopefully it's helpful. If not, you're always free to contact me. We'll talk about that at the end, though. Let's see how it goes first. All right, let's check it out. Okay, so we all know that FSHD is genetically linked to chromosome 4, right? Chromosome 4, and specifically the location 4q35. Okay, so you'll remember that we have 23 pairs of chromosomes, one chromosome each from mom and one from dad, and they're ordered, based on size, and so the fourth largest pair is chromosome 4. And then there's a structure on each chromosome that kind of separates it into a short arm, the P arm for the French word for short or small, which is petite, and the Q arm, which is Q just is the letter after P. And the FSHD region is all the way out the very end of the 4q arm, about the 35th banding, and at 4q35. And that's where the D4Z4 repeat array is. And everybody has a D4Z4 repeat array on both chromosome 4s. And in FSHD, one of them is smaller, right? So FSHD1 is caused by deletions in the this D4Z4 repeat on chromosome 4. Okay, you have between 1 and 10 D4Z4 repeat units. You are potentially FSHD1. The other part, of course, is you need the 4QA FSHD permissive subtelomere. So when you have 1 to 10 repeats on a chromosome 4QA, that is genetically FSHD1. Okay. So you can also have this deletion on chromosome 4 without 4QA. It could be 4QB. This is an FSHD non-permissive allele, and this is not FSHD. And this is actually why the first thing that's done in FSHD testing, or something that it should be done in FSHD testing, is to determine whether or not you have a 4QA or 4QB. If you're 4QB, it just can't be FSHD. So, no 4QA. That is not an FSHD1 chromosome. And then also, you can have 4QA. 4QA is permissive. It is not pathogenic. So you need it for FSHD, but in itself does not cause FSHD. So more than 10 repeats, this is also not FSHD, regardless of whether it's a permissive chromosome or not. Okay, so it's between 1 and 10 D4Z4 repeats, so you can be 2 repeat units, 5 repeat units, 9 repeat units, whatever. It'll be consistent in your family. If you have FSHD1 in your family, everyone in the family will have the same number of repeat units. You're a 5 repeat unit family in general. And in general, the larger the deletion, which means the fewer number of repeats, the more severe, and the smaller the deletion, meaning the more repeats, the less severe, more mild FSHD tends to be. Not always, but tends to be that way. Okay, so why do you need both parts? You need the deletion and 4QA. Well, inside every D4Z4 repeat is part of the DUX4 gene. And the DUX4 gene, as many of you know, or should know by now, the DUX4 gene expression in skeletal muscle is what causes FSHD. Okay? Now, there's only part of it, though, in each of these D4Z4 repeats. The second part of the gene that's absolutely required, the polyadenylation signal, is located in the 4A subtelomere. This is a special signal that allows for the gene to be expressed. It tells the transcription machinery, the gene expression machinery, to put a whole bunch of A's on the end of the RNA. This makes it stable. It's required for protein expression, and then DUX4 protein expression leads to FSHD pathophysiology. So you need this PAS sequence. And so if you're 4B, regardless of whether or not you have a repeat or not, you do not have this polyadenylation signal. So if DUX4 mRNA is made, it is not stable. It becomes degraded. You never get DUX4 protein, and you do not get FSHD pathophysiology. So that's why you absolutely need the 4A as opposed to the 4B subtelomere. All right, so what about FSHD2? Well, FSHD2 is also genetically linked to chromosome 4. Okay, so it's still chromosome 4 because that's where the DUX4 gene is located. But it's also linked to chromosome 18. Okay, so FSHD is digenic. Okay, you still need the chromosome 4 D4Z4 array with a 4A subtelomere, but also you need a second mutation in another gene, most commonly in chromosome 18, in order to get FSHD2. So what's the relationship though? Well, the gene product of the chromosome 18 gene, the SMCHD1 gene, functions to regulate expression of DUX4 and chromosome 4. So they are functionally linked as well. So on FSHD2, the SMCHD1 gene is located on chromosome 18p, so the short arm of chromosome 18, near the end, and you need one mutation, so either the allele from mom or allele from dad, and you need a special D4Z4 repeat unit with a 4A on chromosome 4. Okay, so example of a FSHD2 capable or permissive chromosome. Well, so we used to call this a contraction independent FSHD, but that's not quite true. You don't have that FSHD1 contraction, but you do have to have this kind of mid-range D4Z4 repeat array of between 9 and 20, maybe 22 D4Z4 repeat units, and you absolutely have to have the 4A subtelomere, again, because you still need to make DUX4. DUX4 is what's going to cause FSHD2, and so DUX4 requires a D4Z4 repeat as well as the 4A. Okay, and as we mentioned, FSHD2 is digenic, so it requires two different genes. Okay, so you need this repeat unit and a mutation in the SMCHD1 gene, okay, or a different regulatory gene for FSHD2. Okay, so FSHD2 most commonly is chromosome 18, the SMCHD1 gene. And what is SMCHD1? Well, that is a protein, is a repressor of the D4Z4 region. Okay, essentially this is part of the off switch that keeps DUX4 off, and if you break the off switch, well then DUX4 can be on and be expressed. Okay, so FSHD2 is a little bit different than FSHD1. Okay, there are of course non-permissive FSHD chromosomes. For example, here this chromosome does not have an A. So if you are 4QB, you can't have FSHD1 or FSHD2 or FSHD3, 4, 5, whatever. You can't have FSHD. You need the 4QA in order to make the pathogenic DUX4 gene. Okay. You also can have too many repeat units to be FSHD2. So you might have the SMCHD1 mutation, but if you have 25, 30, you can have up to 100 repeat units, maybe even 120. You can't. Some of those are just too big for FSHD2. We're not exactly sure why. Probably they still get some degree of off, but what we know is those people with clinical FSHD2 tend to have that mid-range repeat size. Okay, so FSHD1 is going to be deletions of the D4Z4 repeat. It's autosomal dominant. You just need one mutation to get between 1 and 10 D4Z4 repeat units with a 4QA, whereas FSHD2, you need a mid-range repeat array. You still need a 4QA, and it's digenic, so you need mutation in the SMCHD1 gene. Okay? So I know some of you, I get questions sometimes, you say, well, I'm not FSHD1 or FSHD2, maybe I'm FSHD3 or 4, and I know some researchers talk about this, and basically FSHD3, 4, 5, they're just FSHD2. Okay, they're just FSHD2 with mutations in genes other than SMCHD1. The DNMT3B gene or the LRIF1 gene, both of these are very rare. They still require a 4QA. So again, if you're 4BB, you're just not FSHD. Okay, so you still need to have 4QA, and you still need a certain size D4Z4 repeat unit. There is one little caveat: the LRIF mutations are recessive, meaning you need 2 LRIF mutations. You only need 1 SMCHD1 mutation, 1 DNMT3B mutation. So this, maybe this one could be called FSHD3. I don't know. They function like FSHD2. Both of these are regulatory proteins of the region. Okay, still digenic, always digenic. Okay, mutation in a regulatory gene. Okay, so this is basically all the digenic forms of FSHD, in my opinion, are FSHD2. Okay, so what we showed, all forms of FSHD are genetically linked to chromosome 4A. Okay, you're going to be making DUX4. DUX4 is on the chromosome 4A. DUX4 causes FSHD in all cases, so it's always going to be linked to that. Okay, but they're also epigenetically linked. Okay, all forms of FSHD have epigenetic changes at chromosome 4A, right? And you've seen some of the videos, hopefully, of these. If not, check them out on what is epigenetics. We can't cover everything in this, but I'll give you a little bit of the flavor. So, epigenetics are the state of the DNA sequence. Okay, genetics is going to be the actual DNA sequence, CTAG, in that order, the sequence of a gene. And the epigenetics are going to be how these are kind of packaged, how a gene can be packaged. So a gene can be packaged in different states. It can be packaged in an on state, and typically this does not have DNA methylation, or it can be packaged in an off state, where this is typically often very heavily methylated. Okay, so in FSHD1, the contracted chromosome 4 is not methylated, and therefore it is in a gene expression state of an on. Okay, the light switch is on. The non-contracted allele is heavily methylated, and you can think of this the switch being off. So you're expressing DUX4 from the contracted allele because of the epigenetics. Okay, what does this mean kind of more functionally? Well, when a sequence is heavily methylated and repeat sequences are often heavily methylated, they become very compacted and contracted. Think of this as a slinky, and I got a video where we show this, where a slinky can be very, pressed together, a spring, and, it's not very accessible, or in the unmethylated state, even though it's a smaller DNA sequence, it can be expanded and able to be expressed. Okay. FSHD2 is a little bit different, because FSHD2, what's broken is the switch to turn things off, not the signal. And so in FSHD2, both of your chromosome 4s are going to be in the unmethylated state, and they're going to be in the transcriptionally on, or the gene expression state. So DUX4 could be expressed from either or both chromosomes. Okay, so that's a little bit different. But essentially, FSHD1 and 2 both have epigenetic dysregulation leading to DUX4 from the array. So we're going to dive in a little bit more to what exactly is the difference. Okay, I've kind of given some of it away, but I know it's always good to repeat things in FSHD, so we'll get to it. So all FSHD is caused by misexpression of DUX4. You just get there by different mechanisms. Okay, the end result's the same, and actually, importantly, at the end, we'll get to this, therapeutically it's the same. FSHD1 or FSHD2, basically the same therapeutic approaches are gonna be applicable to both. You're just kind of getting to the pathogenic gene expression through different systems. Okay, so we're going to start out with a healthy situation. So a healthy, or we'll say non-FSHD, chromosome 4 genetics. Okay, those genetics are going to be that you have more than 10 repeats on both of your chromosome 4. The chromosome from mom, the chromosome from dad. The 2 different chromosomes are just called alleles, if you haven't noticed that yet. And so, this is healthy FSHD genetics. Okay, so let's talk about epigenetics. So it's going to be different. Epigenetics, you have 2 healthy genetically healthy chromosome 4s. And what's going to happen is your genome is going to kind of notice that, wow, these are repetitive sequences. Okay, DNA is repeated a lot in a row, direct repeats on both chromosomes, and it's going to say, whoops, that's no good. Let's turn this off. And actually, your genome has a system, because invasive sequences, viral sequences tend to be repetitive. And so your genome recognizes DNA repeats as saying, hey, these things are not good for the genome. We're going to turn them off. So this is a signal to be turned off. basically what's going to happen is then your mechanism for the cell is going to come in and start loading it up with some factors that are going to say turn this off, okay, and then put on an off switch, an initial off switch on the DNA sequence. It says off, 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 keep this off, and then this is going to come in and say now we're going to keep it off. First you turn it off, and then you want to keep it off forever, and so that's what DNA methylation is. And so you're going to maintain repression or off with DNA methylation, and so both of your healthy chromosome 4s are going to be heavily methylated and off, and this is going to establish an epigenetic environment that is not conducive to gene expression. And the DUX4 gene, remember the DUX4 gene is located right here in this last D4Z4 repeat unit with 4A. The DUX4 gene is located right here. It's methylated, it's off, it's in a bad environment, and it's going to become compacted. Okay, crunched right down. The slinky is going to be pushed together, and this is just going to be off. Okay, so you're not going to get any pathogenic DUX4 gene expression from either chromosome 4. Okay, that's in the non-FSHD state. So what happens in FSHD1? Well, remember, FSHD1 genetics are you now have lost some of these repeats. Okay, you still have the 4A or not. Yeah, you always have the 4A in FSHD, but you've lost some of the repeats. Okay, so this is the situation. So now what's going on is the cell's going to say, whoa, turn this one off, but it's not going to notice this one down here. Down here, it doesn't notice your contracted one. It's not enough of a repeat to signal the cell to shut it off. Okay, so it's just too small. It's roughly around the 9-10 units is where that signal is. Okay, so nothing to see here. Okay, this is perfectly fine DNA sequence, so that's cool. So what's going to happen is you're going to come on, say turn it off. You're going to set up the off switch. Okay, now you're going to come in, try to keep it off, methylate it, and set up an epigenetic environment. But now you see the difference. Okay, only the non-contracted chromosome 4 got methylated and shut off because the cell never saw a signal to shut off the contracted chromosome 4. This did not get methylated, and so this is still on. So you have the switch is off here and the switch is on down here. Okay, so then what happens? You get compaction of one of the chromosomes, and it's really off, and this other one is still on. So you end up getting remember, DUX4 expression is down here. The DUX4 right here, this gene, that's off. It can't be expressed. But the DUX4 gene right here is able to be expressed in your skeletal muscles, and that's how you get FSHD. Okay? So that's FSHD1 epigenetics. In healthy cases, they're both chromosome 4s are heavily methylated and off, and FSHD, one of them is off, and the other one with the contraction is epigenetically on, and the DUX4 gene is expressed. Okay, so basically this is what's going on, and so the system is essentially not functioning right at this locus. So what about FSHD2? Now remember, FSHD2 healthy versus FSHD2 genetically, they actually look very similar. The main difference is right here is the mutation in the SMCHD1 gene, and you don't actually notice this at the FSHD locus. And this is why FSHD1 testing, or a lot of FSHD testing, does not test for FSHD2. If you're measuring the DNA for a contraction, like the majority of FSHD testing does for FSHD1, you miss FSHD2 because at the FSHD 4q35 array, it looks the same for FSHD2 and FSHD and non-FSHD. Okay, so so the key, of course, in FSHD2 is the SMCHD1 gene, and this is an epigenetic repressor of the D4Z4. Okay, and that's something. Remember, we just talked about how healthy chromosome 4s are epigenetically off. Well, that's what SMCHD1 does. It actually helps set up this repression arrangement at the D4Z4 locus. Okay, and so in FSHD2 you have a mutation in the SMCHD1 gene, and there can be different types of mutations that lead to like only part of the protein being made, or maybe a non-functional protein being made, or maybe the whole gene is deleted, and these kind of give you some little different nuances to the type of epigenetic dysregulation, but the end result is DUX4 is going to be expressed and you get FSHD, as long, of course, as you have the 4A FSHD permissive subtelomere. Okay, So, genetically, FSHD2 looks similar to non-FSHD, but epigenetically, it's a big difference, okay? So what's going on? Epigenetically, your genome's going to say, hey, lots of repeats, more than 10, so turn this off, okay? Genome, the signal is there, it says turn me off, okay? Let's keep this gene off. But what's going to happen is the cell can't do it. The machinery that you have to set up and the repressive epigenetics is not functional. So the repeat is recognized to be turned off, but the off switch is broken, okay, due to the mutation of FSHD1 gene. So because of that, you do not get methylation. And this, again, now we pick this up in our epigenetic diagnostic assay. You do not get methylation at either chromosome. So both the chromosome you inherited from mom, the chromosome you inherited from dad, neither one gets methylated. They both are epigenetically on. You could express DUX4 from both chromosomes, and DUX4 expression leads to FSHD. Okay, so what's going on epigenetically? Now you got the comparison, and in healthy chromosomes, they're both off. In FSHD, both chromosome 4s are epigenetically on. And if you have the right other conditions to make DUX4, you express DUX4 at FSHD. Okay, so now if we look epigenetically at FSHD1 versus FSHD2, they both have epigenetic dysregulation. You just get there by different mechanisms, okay? That's what's really important. Okay, in FSHD1, the signal to shut off the gene expression is broken. In FSHD2, the mechanism to shut off the gene expression is what's broken, okay? And so you end up with a situation like this. And so if we're doing diagnostics and looking at DNA methylation, and we look at both chromosomes here, you can see that FSHD1, you're going to see one that's methylated and one that's unmethylated. In FSHD2, they're both unmethylated. Okay, so that's how the epigenetic diagnostic assay distinguishes the two. Okay, now, I told you this doesn't matter for therapeutics, but it does matter for inheritance. Okay, so let's go through the inheritance briefly. So FSHD1 inheritance. FSHD1 is considered autosomal dominant. Okay, it's an autosome, so you have 23 pairs of chromosomes. 22 are autosomes, and then your X, either XX or XY in general, and those are your sex chromosomes. So those are, they're not autosomes; they're sex chromosomes. So dominant meaning you only need to get one mutation in order to get the disease. So in a healthy chromosome 4, you have more than 10 D4Z4 repeat units. In FSHD1, you have between 1 and 10 D4Z4 repeat units. Okay, so for example, 5 repeat units here. Okay, and so now, if you have kids and you have FSHD1, you have a 50% chance of passing it on to each kid. So statistically, if you had 4 kids, 2 kids would have FSHD, 2 kids would not. And these are the different possible combinations with your non-FSHD partner. Okay, and so now often, actually, we see this often skewed a little bit, but it's like flipping a coin. Each time, so you could have 4 kids that are healthy, you could have 4 kids that have FSHD. Statistically, it should be 50%. Okay, these are with FSHD, and you can see, just statistically, you can have either chromosome from mom and either one from dad. Okay, so FSHD2 now inheritance is a little bit different. Now, as we mentioned, FSHD2 is digenic, so you're going to need 2 different genetic mutations in order to get FSHD2, to inherit FSHD2. Okay, so a healthy individual, could have a D4Z4 repeat unit. It actually doesn't matter what size D4Z4 repeat unit, but you could have one that's 15 units, you could have one that's 30 units, but you have normal SMCHD1 copies of the gene. However, a healthy carrier would have a mutation in the SMCHD1 gene, but they don't have FSHD2 because their second component, the D4Z4 repeat array, is too long. In this case, 32 and 30. You need to be less than 20 repeat units in order to have FSHD2. So now to have FSHD2, though, you need both components, the digenic component, meaning the mutation in SMCHD1, and a less than 20 D4Z4 repeat unit on a 4QA. Remember, you always got to have 4QA. allele in order to make the DUX4 gene to get FSHD. Okay, so now how does it affect inheritance? Okay, so FSHD is digenic. That's, sometimes it can seem like it skips generations. Okay, so this individual, if they have FSHD2, they have an SMCHD1 mutation and a D4Z4 that is within the right range to get FSHD2, right? And they can pass that on to a child, but instead of 50%, it's only going to be 25%, okay? Because there's a 50% chance that they get the SMCHD1 mutation and a 50% chance that they get the correct size D4Z4 repeat unit. So that's that's a 25% that they inherit from both from the same parent. Now the other parent actually matters. It's always nice to know that, but they matter because the D4Z4 repeat array can change the math on this. Okay. You also though can have a child that is apparently healthy, and they could be a carrier though of the SMCHD1 mutation. Okay. And so meaning that they have the SMCHD1 mutation for FSHD2, but because they did not inherit the correct size D4Z4 from either parent, they are an asymptomatic carrier of the disease. Okay, and so you also could have healthy children that don't have the SMCHD1 mutation, and if that's the case, it does not matter what size array they actually have. Okay, so now the difficult part is that this carrier, if their partner, if they have children with, has proper D4Z4 size repeat array, like in this case a 17 repeat unit with a 4A chromosome, that could actually lead to FSHD2. Okay, and so that's a case of that there are these carriers of FSHD2 and FSHD2 families that can seem like FSHD is skipping a generation. In this case, it seems like it skipped this generation. Genetically, the FSHD2 mutation did not. It's just because it's digenic. All right, so overall, FSHD1 versus FSHD2, they both result from epigenetic dysregulation and expression of DUX4 from the 4q35 D4Z4. Remember, FSHD1 is caused by contraction of the D4Z4, loss of signal to turn off the gene, where FSHD2 is caused by mutations in the D4Z4 repression mechanism, and it results in loss of the off switch. Okay, so same end result, you just get there a different way. All right, so there you have it. Different mechanisms to get to the same end result, pathogenic DUX4 gene expression. And what this means is that most of the therapeutics, in fact, actually really all of the therapeutics I can actually think of, will be applicable to both FSHD1 and FSHD2. The main reason to know is for inheritance, right? And we discussed that. It's a little bit different, right? FSHD1 is going to be passed on statistically 50% of the time, and FSHD2 is going to be a little bit more complicated, passed on a little bit less. But therapeutically, everything that's coming down the pipeline in the therapeutic development pipelines, they're going to be applicable to both. Okay, the Avidity trial, the Arrowhead trial, Dyne, Myracule, Epic, and all the others that are going to be coming, going to be applicable to both. All right, so I hope you understand now the difference between FSHD1 and FSHD2. And, still, if you have any questions, please feel free to reach out, peterjones@med.unr.edu. Happy to try to answer by email, or we can set up a Zoom call. I do a lot of Zooms with people in Australia and New Zealand and pretty much anywhere in the world where you're watching this. We can figure it out. I'm up at all hours, so happy to set something up. And don't forget to support FSHD Global Research Foundation. Fantastic organization. They really help us out, and they're really helping us with the saliva testing all around the world and finding the FSHD patients everywhere and helping people all around the world. So big shout out and thanks to Natalie Cooney, Emma Weatherly, and of course Bill Moss, who got the whole thing all started. All right, so hope it was helpful, and let me know.